Hi, I'm Tom Metz from Pacific Northwest National Laboratory.
And I'm Crystal Pollitt from Yale University. We're the co-leads of the ChemBio Analytical Sciences Hub of the Network for Exposomics in the United States, or NEXUS, alongside of Randolph Singh from Columbia University and Jeremy Koelmel from Yale University.
Our team is working collaboratively to coordinate community-developed state-of-the-art tools and resources, and to develop best practices to measure the exposome in both biological and environmental samples. As part of the mission of the ChemBio Hub, we are excited to share this educational series with you.
So we're going to be discussing in this tutorial the importance of per- and polyfluoroalkyl substances and give a case for why non-targeted analysis is very important in this field. First of all, we're going to cover per- and polyfluoroalkyl substances chemistry, why it's unique, and why that's led to an explosion of industrial applications and causes toxic effects. We're going to then go into defining PFAS, the different definitions used, and PFAS diversity, followed by a very short history of PFAS manufacture and how it's been released into the environment. Then we're going to go in depth into some of the PFAS uses, what consumer products, how are they used, and those exposure sources. And then we'll go into the argument of why non-targeted analysis is important from the fact that there's a large unknown fraction of PFAS in various matrices, and we'll give some examples. Following that, we'll discuss the differences between targeted and non-targeted analysis, with targeted being the conventional approaches using and showing the advantages and disadvantages of both. Finally, we'll give a teaser on non-targeted analysis applications and interesting findings that will then be followed up in our next tutorial videos. So first, let's go into the unique carbon-fluorine chemistry. So this is a completely anthropogenic phenomena. PFAS is not created in nature. It has not existed until humanity started manufacturing it. PFAS, as far as a chemical structure, consists of carbon with fluorine bonds onto it. This carbon-fluorine bond is extremely strong. So if you look at carbon's electronegativity versus fluorine's negativity, that difference signifies the bond strength. And you can see that fluorine is the most electronegative atom on the entire periodic table, and therefore forms a very strong bond with carbon, stronger than oxygen, nitrogen, hydrogen, so on and so forth. And therefore, this gives PFAS the very strong properties of not breaking down easily. You cannot break them down with normal amounts of heat or pressure. Most enzymes cannot break them down. And so they're extremely stable. Also, because this is a completely anthropogenic phenomena, the PFOA, for example, shown here, which is a type of PFAS, does not mix well with water or oil. It has unique properties, and therefore it's both water and oil resistant. So why the name PFAS, per- and polyfluoroalkyl substances? Per means a fully fluorinated chain, where poly could mean, for example, having CH2 groups or other carbon-hydrogen moieties. But they both have high levels of fluorination. And then there's also polymers. Polymers are something that's not going to be covered by our measurement techniques of mass spectrometry, and they're usually inert, so they could be assumed to be less toxic, although additives or breakdown products, such as in plastics, the same concepts, could lead to exposure. Now, if we look at the bottom, we can see perfluorooctane sulfonate, so that's PFAS, and you can see the head group and the carbon-fluorine chain. You can also see perfluorooctane carboxylate, and then you can see the CO2 head group and the chain, called the tail here. So this is called a subclass of PFAS. So PFAS is not one molecule, it's a group of many molecules. And the subclasses consist of one head group, or it could be multiple functional groups, followed by varying lengths of the chain. So you can have sulfonic acid with 1 carbon fluorine or CF3, 2 CF2 chain length, 3 CF2 chain length, etc., etc. And all of these have slightly different properties. Now, how do we define PFAS? We talked about the overall concepts of what PFAS are and their definitions, but there's further classifications to narrow down databases. So for example, PubChem can have about 6.5 million PFAS in their database as defined by having 1 saturated CF2 or CF3 part. Although if you look at molecules that have at least 1 fluorine in them, there's 20 million compounds of fluorine attached to a carbon. On the other hand, let's say we're interested in regulation, then there's only 27,000 PFAS in the PubChem database that meet this criteria. And therefore, it's important to think, what is the purpose of my definition? Am I interested in regulation? Am I interested in the health effects of PFAS? So on and so forth. Then we have Comtox Chemicals Dashboard. So this is a different database. There's a few databases that have PFAS structures in them. The Comtox Chemicals Dashboard has about 15,000 PFAS compared to the millions of PFAS in PubChem. So you may think, well, then I definitely want to use the PubChem database because it has more coverage. Well, this might not be the case. So Anthony Williams and other team members at the EPA have manually curated the PFAS in that EPA dashboard and reviewed them to make sure that they're PFAS that likely exist in the environment or have been synthesized. On the other hand, PubChem has PFAS, for example, from patent literature or from chemical synthesis companies, where they just generate all the possible structures of carbon fluorines that could even exist chemically and put them into our database so that they can sell them if you look them up, even if they've never synthesized them. And so therefore, a significant portion, let's say a majority of the PFAS in PubChem don't actually exist in nature. So for our purposes, measuring the environment and exposures, we want to focus on the Comtox Chemicals Dashboard or narrow down our searches in PubChem to those that actually may exist. So we talked about the OECD definition. In Comtox, the current definition is 30% fluorine by atoms excluding hydrogen. So this is a really nice definition because it gets to something being more fluorinated than not. You could have something that has a CF2 group but has hundreds or thousands of atoms, and that's just a small portion of its chemistry. So this is getting to the point of what is mostly fluorinated, and hence what has mostly PFAS properties, both in the environment, human health, and for industrial applications. So I like this actionable definition. They also had another definition previously of containing the following substructures. So we can see that there's not quite a harmonization on how PFAS are defined, and obviously this is important from a regulatory standpoint, but also from a non-targeted standpoint or from a measurement standpoint and a human health standpoint. Where do we focus our resources? What do we consider a PFAS and not a PFAS? Here's the PFAS timeline. So in the 1930s, Teflon was accidentally discovered. It was specifically 1938, and then it was used in significant amounts in the 1940s in the Manhattan Project for the development of the atomic bomb. And so there were scientists who had a lot of experience with the perfluorochemistry, and then in 1950s, Teflon was used in consumer and industrial products, starting with a pan, where the wife of one of the scientists working with PFAS asked whether they could put it on a pan and what would happen, and they saw these amazing non-stick properties. So there are non-stick pans that we still have up to today, and so that's where it started to be born into use as a consumer product. Then in the 1960s, there was AFFF, or aqueous film forming foam, and this was used to put out fires. It worked much better than other technologies, and so it's now become the gold standard, and this is almost fully perfluorinated alkyl substances within that mixture used to fight fires. Then in the 1970s, there was an explosion of the use of these different chemicals. So you can think of Gore-Tex, raincoats, rugs, and aerospace, and solar panels, and drugs. So a lot of different applications of these PFAS. By 2000, there was a global distribution of certain PFAS, found everywhere from the Mariana Trench to Antarctica. There was a voluntary phase-out of products. Voluntary, of course, because there was public, scientific, and legal pressure on 3M specifically to phase out these products. That led to increased public scrutiny as there was more publicity about the health effects, new research and scientific discoveries, a lot of grant funding going towards this, and on the other side, beyond lawsuits and alerts, was the manufacturing base started to spread out. So from 3M and a US mainly centric industry, it moved to China and other countries with different regulations, and some of them lacking regulations altogether, to synthesize these chemicals for consumer use. Also, the diversity of the different types of PFAS continued to explode. So in order to have use in many different applications, there has to be a lot of different types of structural moieties. Some of them might be better for surfactants, some might be better for coating, some might be better for their strong durability or lubricants. Some may be better in food packaging to be more food grade, quote unquote. And so we have a large diversity of PFAS structures. Here's just an example of some possibilities of PFAS. Note that there is a head group, like we talked about previously for each PFAS, and then a repeating chain. So here's a commonly used graphic showing some of the applications of PFAS. Everything from pesticides, cleaning products, dental floss, nonstick cookware, rain jackets, rug, pizza boxes, and other takeout containers, paints, sealants, varnishes, construction materials. So the list goes on. Cosmetics are a big one. And so the point here is that it is very hard to avoid exposure to PFAS, and that in order for PFAS to be used in all these different consumer products, it needs many different chemistries, leading to a very diverse range of PFAS chemicals we're being exposed to, all of which might have different toxicities and fade and transport properties in the environment. So like we talked about, PFAS is everywhere. If we imagine our house, we could look at our furniture, and so there would be flame retardants, also brominated, chlorinated species, but also PFAS. We could look at food packaging, cooking, showers, because it gets into our water system, which we'll discuss from many different sources, cleaning products, paints, carpets. And so we've done a lot of studies on PFAS, and we find that, for example, house dust has an extremely diverse range of PFAS, hundreds of different PFAS molecules at very high concentrations. And so we are being exposed to high levels of PFAS in the home because of its use in all of these consumer products and it entering our water and food. This finding was actually interesting because the house dust had even more concentrations of PFAS than other sources such as road dust or even urban dust outdoors. And so really this is an indoor exposure as one of our major sources, although we are exposed in various other places such as in our cars and outdoors. Here's an example of some more structures of PFAS. Highlighted in the square are PFOA and PFAS. These are the usual PFAS measured and originally were measured and phased out. But you can see that they're just a minimal subset of the 14,000 plus PFAS that can exist. So what about sources, transport, and fate of PFAS in the environment? Here you can see that there are many different sources of PFAS, and we'll zoom into these different sources. So let's start with airports. These can be a major source of PFAS and create local hotspots. So there are hotspots, even though PFAS are found everywhere. There are places where people are exposed at much higher levels, and the health effects that they experience can be much more significant. So PFAS, for example, in firefighting foam, is very high. It's what firefighting foam is mostly made out of at airports, both due to training exercises and to actual fires and to accidental spills from hangars that have to be filled with large amounts of firefighting foam. There are commonly large emissions of PFAS into the local waterways, soil, etc. I think an interesting side note here is that, for example, in Japan, United States bases, which have a lot of Air Force activity, are the major source of PFAS in Japan because of the high use of AFFF. Another source of PFAS is landfills. So landfills, as we talked about, consumer products are of high levels of PFAS, and landfills are going to contain this large diversity of PFAS molecules. Now, these PFAS molecules are going to leach from those consumer products in the landfills into leachate, which is just garbage juice. Now, a lot of landfill operations have ways to collect that leachate and treat it before that liquid is released back into the environment, but these treatments do not remove PFAS. So, for example, transfer stations, other spots along our refuse chain can emit PFAS. And we talk about PFAS diversity. It's not just what the manufacturers produce, because they produce certain PFAS. There are byproducts they produce they don't even know about. They emit these into the environment, but more importantly, these go into consumer goods. We use these consumer goods, and then they go into the environment. So especially in landfills, where there's very corrosive, acidic environments, which lots of microbiological microbiome activity, there is a breakdown of these PFAS, so what we call precursors, into their terminal products. So here's just a fun movie of Nicole, and this is research done at the University of Florida, where John Bowden's lab is one of the premier research institutions looking at PFAS in municipal waste. Now, these PFAS, for example, PFAS and PFOA might be phased out, but these can actually be terminal products. So while the manufacturers are producing other PFAS, they end up mostly turning into PFAS, PFOA, and other similar molecules. So just because one molecule might be regulated at the manufacturer side doesn't mean that it won't occur later down the stream due to production of other molecules. Another source, as far as refuse goes, is sewage spills. So, for example, here is a map, also work done by the University of Florida under John Bowden's group, looking at thousands of surface water samples and the distribution of PFAS in Florida with this high-resolution map, looking at surface waters throughout Florida. And then if you look at the reported sewage spills in Florida, you can see that they map almost identically to PFAS, and we know that sewage has high levels of PFAS in it. Also, if you look at the population density of Florida, that also maps with where sewage spills occur and where PFAS exposure is highest in terms of surface water levels. And so population dense centers may be a convoluted parameter with sewage spills, but you can see that there are hotspots linked to populations and sewage release. Now let's get into our next question, and our next question is going to be, why do we do non-targeted analysis? So we'll continue to talk about some of these sources in the environment of PFAS molecules, but now let's focus a little bit on what these PFAS molecules are in terms of what is covered and what is not covered by traditional measurement techniques. So if we look at wastewater and sewage, which we just discussed as a major important factor in PFAS exposure, you can see that a targeted method in black with 52 PFAS covered only shows less than 90% of the PFAS burden. Now, this PFAS burden can often be determined usually using extractable organic fluorine or other total organic fluorine methods that look for all of the organic fluorine, even if we can't determine their structures. So we can see that there are a large amount of short chain. So these are C2CF3 or C3, so 3 or 2 carbon fluorine containing species, medium chain, and some long chain PFAS species. And then there are quite a lot of unknown PFAS. If you look in the green and the gray, these are all unknown PFAS molecules, and therefore what we can assume is that most of the PFAS is not covered by traditional techniques. Now note that some of this fluorine, organic fluorine, may be from sources we don't care about in terms of our PFAS definition, depending on what that is. So some of them might be pharmaceuticals that just have a single CF3 unit, so on and so forth. So there is a little more work that needs to be done to determine what actually is the unknown PFAS burden, but this does give an idea that most of the PFAS we are missing, whether those be polymers, other PFAS we can measure via mass spectrometry, etc. So another PFAS source is public drinking water. Again, just like the leachate and the sewage that we just discussed, most of the PFAS is not treated for, and so it continues to cycle into the system. So for example, in areas such as California, where water is strongly reused, which is a good thing, there's water scarcity, a lot of that PFAS that enters the water system through sewage, through our sinks, showers, etc., it then cycles back into the water treatment plant, is not treated, and starts to concentrate in this cycle. Another source of PFAS to water and air is the manufacturing sites themselves. So for example, we can see here, this is one major manufacturing site, let's say 3M, DuPont, Chemours, Solvay. There's some major companies both in the US and abroad that manufacture, and a lot of them discharge directly into surface waters or have illegal disposal depending on the country, or are within legal means, have some type of filter before they output into the water systems, but do not remove all types of PFAS. So we're working very closely with the Environmental Protection Agency, Jackie Bangma, Mark Striner, and James McCord, and they've developed a wonderful workflow where they are able to go to the sites, the manufacturers' sites themselves, which they did about a decade ago, look at all of the different effluents, both treated and non-treated, coming out of some of these manufacturing sites, and then use FluoroMatch which we'll discuss in other tutorials, which is one of the PFAS non-targeted software we're developing to be able to curate and annotate these compounds using non-targeted analysis approaches. So the take home here is that using non-targeted analysis, they're able to identify these new PFAS molecules and then build an encyclopedia of all of the PFAS that may exist. We can think of this as the fire. The industrial site is the source of that fire, and everywhere else is the PFAS are coming from this as the point of origin, whether they're manufactured and put into consumer products, whether they're put into lubricants. The manufacturer site is the point of origin where that diversity can be originally characterized, and then these PFAS can be looked downstream. And so we have this now encyclopedia that we continue to build of all these PFAS molecules. Jackie Bingham, Mark Striner, and James McCord are the people from the EPA. Rachel Smolinski also worked at the EPA, developing these libraries, and Parker Chain from Yale University, from the Crystal Paulett Lab, also developing these libraries for eye mobility. So, what about known PFAS across time in human serum? Since the early 2000s, there has been a lot of pressure to reduce the levels of PFAS in the environment. And as you can see, that pressure actually worked, especially the legal pressure, with many multi-billion and even dollar lawsuits that these companies have lost. So you can see some of the commonly measured PFAS decreasing, especially PFAS and PFOA, over time till 2016. Now, this is the NHANES cohort. We don't have up-to-date data in human blood in such a large cohort in the United States since then. And so this is a gap in the data since this is a decade old. But the idea is that PFAS, PFOA, and other PFAS are decreasing. Note these are the commonly measured PFAS. Here's another study looking at Sweden showing the exact same thing over the same period, is that PFAS were continuing to increase in human blood, but then in 2001, due to pressure, started to decrease dramatically. So a very good finding that's exciting to know how political, legal, scientific communities can get together and work to lower the burden of this toxic chemical in our blood. But here's another plot showing a very different trend when we look at things slightly differently. So if we look at extractable organic fluorine, again, that idea of looking at the total fluorine or PFAS burden, we can see that while there are some high points prior to 2000, the general levels of this in the blood do not follow a downward trend. In addition, the targeted only showed 6 nanograms per gram of PFAS in the blood when you sum all the 21 targeted PFAS, but when you looked at extractable organic fluorine, 30 nanograms per gram were measured, which shows an 80% unknown. So, what this shows is that the known PFAS are decreasing, the total PFAS are staying the same, which means the unknown PFAS are increasing. And so this is an important lead up to why non-targeted analysis is important. Unknown PFAS must be increasing in serum. Measured PFAS are only a fraction of total PFAS. We need to know what these PFAS are, how much, how toxic are they. The EPA has shown that different PFAS with different structures can have very different fate and transport properties and very different toxicities. We need non-targeted mass spectrometry and software to be able to characterize these chemicals, since targeted approaches miss the majority of PFAS. So here's an example from the University of Florida showing the different levels in which we can analyze PFAS. So using target analyses, we may be able to measure 40 PFAS. A lot of labs measure between 30 and 40 PFAS, but as they add to their panel, it can get 200 or even 200, maybe 300 max PFAS molecules. very expensive because you have to buy chemical standards to do this targeted analysis. You have to know what you're looking for. Targeted. Non-targeted analysis, on the other hand, you can cover quite a bit more PFAS because you're using universal principles of chemistry to try to characterize PFAS using mass spectrometry data without having to know what those PFAS are beforehand, or even for them to be in a database. You can completely de novo characterize PFAS you haven't seen before. Also, as companies release new PFASs into the environment, some of which they may not report, we can determine what they are. Then there's about 14,000 PFAS, let's say 15,000 in the EPA database. Now, even with non-targeted analysis, it has to be amendable to our instrument. That means able to be measured, and therefore not everything's able to be measured by our instrument. Not everything is going to be viewed in the specific extractions that we do to prepare those samples. Some things may be lost while others are concentrated so we can see them better, or some might just be too low in abundance to be able to be detected. And then, of course, we have our total organic fluorine, which is even a bigger realm. Maybe things that even don't exist in the database, might not even be PFAS, so a much larger portion. So looking again at another study, this one was done on surface water, and so taking that point home, that using LC high resolution mass spectrometry or targeted triple quad approaches, only about 2% of the extractable organic fluorine in these surface waters all across North Carolina could be viewed. So most PFAS we are not detecting. So what's the difference between non-targeted analysis and targeted analysis? Targeted analysis, you have an ease of analysis. You have a synthesized chemical standard. You can be very confident in your results. It's faster. For example, the data analysis may be able to be faster. It has lower cost. It uses triple quads, for example, instead of high resolution instruments in many cases, which cost significantly less. It also takes less expertise to run and to look through the data, so the personnel costs are lower. It's reproducible. Between labs, you actually get a reported concentration. So, for example, nanograms per gram in blood, as we discussed, and those can be comparable between labs. It's hypothesis driven. We have to know which PFAS we're looking for, and we can specifically target those. There's ease of interpretation. Often we know more about these PFAS molecules, such as PFOA and PFAS, and their toxicity, so the levels we measure have actionable interpretation, and as I've mentioned, it's quantitative. You actually get a concentration. Non-targeted analysis, the big advantage there is comprehensive coverage. So there is some ease at suspect screening, which is a hybrid where you can look for thousands to tens of thousands of chemicals that have spectra available, or at least we know their fragmentation. So for example, in FluoroMatch, which is our non-targeted software, it has over 10,000 chemical species with their fragmentation, and you can readily start identifying this, and still very confidently, versus targeted, which is just, 100, maybe a few hundred at most. You can also discover novel chemicals, and this is where non-targeted analysis comes in, using fragmentation patterns and other pieces of evidence we'll discuss in further tutorials, that piecing all these together, you can discover unknown PFAS. Non-targeted analysis is fun, so it's extremely exciting to be able to discover all of these new molecules. There's no bias in the question, and then there's system-wide understanding. So PFAS are everywhere, and they're in innumerable forms. and we need non-targeted analysis to cover those. And so we've started to develop, both prior to and part of NEXUS ChemBio Hub, tools to be able to characterize these PFAS in the environment. And here you can see some examples. So industrial effluent, like we talked with the EPA, we were able to identify over 1,000 PFAS molecules in a single industrial effluent, something that could never be done by extractable organic fluorine, which just gives you total PFAS, or targeted methods. And so we can now actually know these structures, look at them downstream in chemical products, in consumer products such as cosmetics, blood, in garbage refuse and leachate coming out, in firefighting foams, and in biological organisms, and looking for their products as well as their transforms. So we can now trace these PFAS all across the environment. And that's important because, as we mentioned, each different species can have different toxic effects, and therefore it's needed to look at these different species. We also have the fresh air wristbands, and these wristbands can pick up PFAS in the air, so we can actually do exposure monitoring of airborne PFAS, which is a matrix of exposure that has very limited knowledge on what those types of PFAS are and the exposure dynamics there. So now with non-targeted analysis, we can start building a better picture of exposure dynamics. So in conclusion, PFAS are diverse, both in terms of their spread across the environment in consumer products and in their chemical structures, and it is essential to understand this diversity to understand that impacts on human health. This diversity is not currently covered in conventional approaches, and 3, we need non-targeted analysis to help fill that gap. Please see next videos for state-of-the-art approaches for non-targeted analysis and how we can actually fill this gap in these technical tutorials following this.
Thank you for watching. If you have any questions or suggestions for future topics, we welcome your ideas. To stay informed about the latest developments in exposomics, please visit our website and subscribe to our monthly newsletter from updates from across the field. Be sure to explore the NEXUS podcast on YouTube and Spotify, and follow us on LinkedIn, X, and Blue Sky and NEXUS Exposomics to stay connected with our community. Thank you for being a part of our NEXUS network.